Welcome to your lecture on hearing. Throughout this lecture, we're going to cover a lot of information, and I'll be relating it back to phototransduction. While phototransduction occurs in the eye, they're good parallels to make when we're talking about acoustic transduction. So the first thing that we want to talk about is transduction. Transduction is the conversion of a sound wave into a neural impulse. The structures involved in this are the hair cells of the inner ear. Now, when we're talking about transduction in the eye, we talk about the retina and we talk about the photoreceptors, the rods and the cones. Here in the ear, we're going to talk about a structure called the cochlea. And in the cochlea, there's going to be conversion cells called hair cells. So the hair cells are kind of like your rods and your cones. The cochlea is kind of like the retina. The cochlea is the overall structure. The hair cells are the specific cells that do acoustical transduction. With hearing, you want to understand kind of what makes up the idea of our sense of hearing or our sense of audition. The three components. The first is frequency. So that's whether or not something has a high pitch, a low pitch, or a medium pitch. High pitch, think very shrill, squeaky voice. Low pitch, think very deep, almost kind of baritone voice. The intensity. You can have someone who has a very shrill voice that is screaming, or you can have someone who has a very shrill voice and is whispering. So intensity is going to be how loud something is. And the last one is quality or timbre. Now, quality or timbre is what allows us to decide where a specific pitch with a certain intensity, what it actually came from. And so the easiest way to kind of think about this is that different objects actually resonate different qualities of sound. So if you were to look at, say, any instruments that are similar in nature, a guitar and a violin, they both use metal strings. However, the guitar makes a very unique sound in comparison to the violin, even if it's just the plucking of the string. We saw this slide back when we were looking at vision, and I told you before, the length of the wave indicates the hue or the color. So from peak to peak, if it's a short wave, it's the small arch of the rainbow, and you're going to see your violets or your blues. If it's a really long wave, you're looking at the top arch of the rainbow and you're getting your reds. And again, the length is in nanometers. So small number of nanometers, shorter in length, long number of nanometers, longer in length. The same thing holds true for pitch. Pitch being how high or how low we perceive a sound to be. Not how loud or intense, but literally, is it high pitched or low pitched? So if something has a high frequency, if the waves are very short and compact, that frequency of wave gives a high-pitched sound. However, if the waves are kind of slow rolling like you see here in the red and far apart, then that's going to be a lower pitch. How loud a wave is determined by how tall it is. So amplitude, the height of the wave, indicates whether or not it is a loud wave or a quiet wave. When we talk about it in colors, a tall amplitude means a very bright color, or you could even say a loud color, where a short amplitude is going to give you more of a dull color. Same thing with sound, and I always like to go to the beach and think this way. If you're at the beach and 10-foot waves are rolling in, it's going to be loud. If you're at the beach and a 1-foot wave comes rolling in, it's going to be a much softer, less intense sound. It's important to note that not all sound is created equal. In fact, loud sounds do cause hearing damage. So as we look at sound in decibels, anything above 85 decibels can start to produce hearing loss. The world that we live in right now has a lot of noise, and that impacts our hearing. So what we are seeing is that younger and younger generations that have greater technology that pumps sound directly into their ears, they're actually showing increased hearing loss at a much younger age. We talked about quality again, and if you go back to your textbook and read through it, they'll tell you the exact same pitch and the exact same intensity played by two different instruments. The fact that you know one came from a guitar and one from a zither, or even a guitar and let's say a trumpet, they played the same note at the exact same intensity. The fact that you can distinguish what object it came from, that's the quality. For the ear, just like the eye, you need to know the different structures. The ear is organized into three sections: the outer ear, the middle ear, and the inner ear. Pause the video right here to write down the different structures. If you have the opportunity, please try to click on this link and see if you can pull up the video on hearing. It talks you through the entire process structure by structure. But one of the more important things for us to recognize is that if we are looking at the interior structure of the ear called the cochlea, The high-pitched frequencies are processed at essentially the outermost part of the cochlea, and as it winds deeper and deeper in, lower pitched sounds are processed there. Now I'm going to talk you through the different structures. First, the outside part, the ear that you can actually put your earring through or the jewelry on, that's called your pinna. The pinna acts as a satellite and it collects sound waves. Those sound waves are then channeled through what's called the auditory canal. And again, both of these structures do what we call conduction. They conduct a sound wave. The sound wave then impacts what's known as the tympanic membrane. The tympanic membrane literally is a membrane that stretches across the auditory canal and vibrates. We often call this the eardrum. The tympanic membrane starts the middle ear section. The tympanic membrane vibrates, and this vibration causes the motion of three bones in the ear called the ossicles. They impact each other, and ultimately the stapes, or the stirrup, is going to act like a piston, and it's going to actually push on another membrane called the oval window that is part of the cochlea. Once we start talking about the cochlea, we are now talking about the inner ear of hearing. And so the inner ear is going to consist of the cochlea and its semicircular canals and the auditory nerve. The cochlea is like the retina in the eye. This is a structure that does acoustical transduction, turns a sound wave into a neural message that the brain can understand. The cochlea is lined with something called the basilar membrane. The basilar membrane causes a stimulation to the hair cells that are attached to it, and those hair cells, like the rods and the cones of your eye, do acoustical transduction. They turned a sound wave into a neural message. If we were looking at which ones deal with the conduction of a message and which ones deal with transduction, the pinna, the auditory canal, the tympanic membrane, the ossicles, those are all going to be considered conduction structures, even up to the oval window. Once we use the term cochlea, we are now talking about a structure that does acoustical transduction. It's involved in the transduction process. And the auditory nerve is going to send the message. The semicircular canals are also these fluid-filled bony labyrinths that come off of the cochlea. Their job is to help you with balance. Sometimes we'll hear people say they have an inner ear problem and that's why they have a hard time maintaining balance. If that's something someone says, it's because they're dealing with issues of fluid regulation in their semicircular canals. The cochlea is the structure that you need to know best. So, it's a bony, fluid-filled tube of the inner ear that does acoustical transduction. It changes a sound wave vibration into a neural signal. This happens when fluid in the inner ear of the cochlea is being stimulated by the pumping or the action of the stirrup. It causes fluid to flow through and around the entire interior of the cochlea. This flowing of fluid causes hair cells of the basilar membrane to actually move back and forth. The moving back and forth stimulates the attached nerve cells, and those impulses are sent out the auditory nerve to the temporal lobe of the brain. Obviously, the first stop off will still be the thalamus, so it can register it as an auditory message. The basilar membrane runs the length of the cochlea and it holds the receptor cells, which are the hair cells. The picture here is of a hair cell, and it's much more like a bristle-like hair as opposed to the hair that you might have on your head. But stimulation of these cells causes the change from a vibration to a neural impulse. The auditory nerve then carries that information first to the thalamus and then ultimately to the temporal lobe of the brain where hearing is processed. Just a close-up look at the cochlea. And again, please remember high-pitched sounds are going to be processed at the beginning of the cochlea, and as you work your way deeper into the interior of the cochlea, that's where your deeper sounds are going to be processed. Sound waves hit the tympanic membrane, cause vibrations throughout the three bones of the middle ear, your hammer, your anvil, and your stirrup. The stirrup moves in a piston-like motion, pushing fluid through a ascending aqueduct and then through a descending aqueduct, but that's a little bit more than what we really need to know. And that motion of the fluid causes the hair cells in the basilar membrane to vibrate back and forth, causing a neural impulse that will eventually get processed in the brain. Here is your auditory cortex of the temporal lobe. Again, that is going to be your primary auditory cortex. That is going to be the end place for all of these neural impulses to be understood. If you have a chance, here are some hearing illusions that you can definitely click on and enjoy. But hearing, just like vision, is about perception. We filter out much of it and focus on some of it, and often our vision will misconstrue what we think we're hearing. You need to know two theories for how we perceive pitch. So the actual pitch of something, how high pitched or low pitched it might be, how high the information or how low. Now, place theory says that if we were to unroll the cochlea, and kind of think of the cochlea as rolled up like a Cinnabon, if you were to unroll it, that across that entire basilar membrane, across the entire length of the cochlea, very specific points are going to have very specific pitches. Now, the problem is with this is that it doesn't really work that way for deep tones, but for high pitched sounds, it does to a certain degree work that way. It has the kind of way to remember this is that it acts like a harp. And when we say that it acts like a harp, if you've ever seen a harp, harps have about a couple hundred strings, and every note has its own unique string. So place theory says that each of these hairs acts as its own kind of indicator. It's its own way of determining what pitch you hear. Now, again, partially right. It's right for things like high pitch, but not for the deep process pitch tones. The next theory of pitch is what we call frequency theory. And frequency theory basically states that instead of it being very specific places, that it is in fact going to be the entire basilar membrane vibrating at a certain frequency. So if it's high pitched, it's going to vibrate at one rate. So here we have it at 100 hertz, and it's going to vibrate, sending an action potential up the auditory nerve at a very specific rate. But let's say we make it an even higher pitch. We bring it up to 200 hertz. It vibrates much faster and therefore more frequent action potentials. Now, both of these have some part that is correct and they have some parts that are incorrect. For the frequency theory, we do know that the basilar membrane does vibrate, and it vibrates somewhat like a drum, where certain areas of it will vibrate together. But we also know that it doesn't vibrate at a rate high enough to meet some high-pitched sounds that we can hear. As I told you before, both theories have some flaws, but both have some valid points. Helmholtz theory was correct, except that hair cells in the basilar membrane weren't actually independent. They vibrate together as suggested in frequency theory. And waves don't necessarily peak at a particular place. Also, like I said in the last one, neurons fire at a maximum rate of 1,000 impulses per second, but we have some sounds that we hear at 4,000 hertz, and so we use what we now think is the volley principle to explain that one. Ultimately, what our current thinking is for pitch is that both place and frequency theory happen in the basilar membrane. Low frequency tones appear to be translated into pitch theory through frequency coding, but the high pitch might actually have to do with more specific locations on the basilar membrane. So depending on the pitch and depending on what's going on, it can be a little bit from column A and a little bit from column B. So it's important that you understand both theories of pitch. Frequency theory, the rate at which the entire basilar membrane vibrates, generating an action potential or a neural impulse that's going to go up the auditory nerve. Place theory, that there are certain locations on the basilar membrane that will actually perceive specific pitches. We also do something called localization of sound. This means how can we locate a sound in space? And really it's two things that allow us to localize sound: intensity differences and time differences. Intensity differences. If you have a sound coming from the left side of your body, your left ear is going to hear a much louder sound than your right ear. So we know that it's probably coming from the left side. But also you have this lovely head right between your two ears, and it acts as a shadow or a partial barrier. So there should be a time difference. So the time for the sound waves to hit your left ear should be much faster than to hit your right ear, and therefore between the time difference and the intensity difference, your brain can make the calculation that the object or the sound came from the left side of your body. Again, here's just another video where we can find out that vision can actually conflict with our hearing, and usually vision wins. Last little bit that we have to cover has to do with hearing loss. There are two types that you need to know: conduction and sensory neural. Conduction hearing loss deals with any structure that we decided had to do with the conduction of a sound wave. That means every structure up until you get to the cochlea. That includes the pinna, the auditory canal, the tympanic membrane, and the ossicles. Damage to those can either cause deafness or some hearing loss. If you ever had damage to the tympanic membrane, it could heal itself. It might heal itself and be scarred. Scar tissue does not vibrate the same way that a non-damaged tympanic membrane would vibrate. And in that case, you might not hear as well. So the conduction of the sound waves isn't as efficient as it once was. You have conduction hearing loss. Sensory neural hearing loss has to do with everything that might be damaged to the cochlea's receptor cells, those hair cells, or to the auditory nerve itself. And we often call this nerve deafness. Now, you could be fully deaf, say your auditory nerve is damaged, or you could have some hearing loss. So let's say you listen to loud music and it ends up causing some of the hair cells in your ear to break. Those cells do not regenerate, and therefore that area that would be responsible for that amount of hearing may actually show that you start to lose your hearing. As you get older, you will start to not hear high frequencies as well, but we maintain the low frequencies. Another way to think about this is the high frequencies are processed at the very kind of front of the cochlea, not deep inside. So they're probably impacted by the most waves, causing the most wear and tear, which might indicate why we get rid of those or don't hear those sounds as well. In class, I gave you guys exposure to the mosquito. And again, this is just a piece of technology that utilizes sound waves against us, and it uses high frequency waves to annoy teenagers. And those waves are ones that adults, because of natural hearing loss, do not perceive. The last thing that we will cover all together is cochlear implants. And this is just a way to restore hearing in somebody who may in fact have some form of deafness or loss of hearing. For a cochlear implant to work, there must be a functioning auditory nerve. Now, this just kind of hits home on the entire idea that hearing is about a combination of really electrical impulses that go to the brain and the brain's ability to put them into a meaningful pattern. The cochlear implant is an exterior microphone processor. It picks up or amplifies sounds from the environment, and it sends that information through a catheter that is wound through the cochlea. The catheter generates an electrical impulse, which is picked up by the cochlear implant and stimulates the auditory nerve. Those impulses then travel to the thalamus, and then from the thalamus to the brain, and the brain then puts together the pattern as an indication of sound. Okay, so if you guys have any questions or any concerns, anything that you need me to go over again, please let me know. You should be really good with things like the structure or the anatomy of the ear, what those structures' functions are, how acoustic transduction happens, and the two theories of pitch. Generally with hearing, there's not as many moving parts as with vision, but you should still be really good with pitch theory, localization of sound, structure, function. Again, any questions, please let me know.